fungi. Fungi are eukaryotes, just like animals and plants. Although they have the same basic genetic structures as do animals and plants, there are some differences arising from the fungal lifestyle that need special explanation, which is one reason why they merit special study. There are other reasons that make fungi worthy of investigation in their own right, however, which we will explain in the fall. The part played by the fungi in the nature make them crucially important to the maintenance of life on Earth. Secondly, they are enormously useful to use in industry right now, and they have an enormous potential for us to exploit in the future. As probably the oldest line of eukaryotes, they provide us with easily studied model organisms. Yeast are favorable organisms for study of genetic phenomena. The yeast that has been most studied is Saccharomyces cerevisiae. Perfect yeast which can reproduce both asexually by budding and sexually by ascospores. Haploid cells of Saccharomyces cerevisiae have 17 chromosomes. Yeast contain also plasmid. Many have two mating types considered analog to male and female E1A-. Can grow vegetatively upon mating. A diploid cell is formed. Saccharomyces cerevisiae is the model. It's believed to have been originally isolated from the skin of grapes. It's one of the most intensively studied eukaryotic model organisms in molecular and cell biology, much like Escherichia coli as a model bacterium. And this is its colony, and this is appearance under microscope. Saccharomyces cerevisiae was the first eukaryotic genome to be completely sequenced. The genome sequence was published in 1996 and has been updated regularly in Saccharomyces genome database. Currently, it's considered that the genome is composed of this numbered base pair and 6,275 genes organized on 16 chromosomes. The ability to culture this yeast species as haploid simplifies the isolation of mutant and haploid-diploid hybrids. The ability to culture this yeast species as haploid simplifies the isolation of mutant and haploid-diploid hybrids. Saccharomyces cerevisiae is a small single cell with a doubling time of 1.252 hours at 30 centigrade, and importantly can be cultured easily. Consequently, they permit the rapid production and maintenance of multiple strains at low cost. Saccharomyces cerevisiae has been developed as a model eukaryotic. Production of fermented beverage. Its use. Production of fermented beverage and breads, spoilage of food and beverages, processing food waste, production of food ingredient as probiotics, production of industrial ethanol, and others.